Hello, everyone, and welcome to The Current. This is the North Central Region Water Network's Speed Networking webinar series. My name is Rebecca Power, and I am joining you from the University of Wisconsin-Madison Division of Extension, and I will be moderating today's session. Since some of you may be new to the North Central Region Water Network, we are an extension-led collaboration among land-grant universities and our excellent partners in 12 upper Midwestern states. Today's topic, we have a diversity of research and outreach programming presentations around groundwater and nitrates from across the North Central region. Much of our region relies on groundwater for drinking and, of course, for other uses. And we'll hear about a couple of different research projects as well as some extension programming from right here in the state of Wisconsin. Before we introduce you to our speakers, just a few notes for you participants about the way that these sessions work. First, we'll be running through all three presentations, and while we do that, you can submit your questions for presenters in the chat box. The chat box is accessible through the purple Collaborate panel. There's a little tab in the lower right-hand corner of your webinar screen. And we'll keep those questions for a dedicated Q&A session at the end, and we try to leave a good amount of time for that. If you're having issues with audio, there is a phone-in option, and that can be accessed by opening the session menu in the upper left-hand area of your webinar screen and selecting use your phone for audio. And then also for those of you that are joining as participants, please make sure that your microphone is muted when you join. That will help everybody out. And we'll send a note in the chat box if we need to remind folks of that. This session will be recorded, and you can access that recorded session at northcentralwater, all one word, .org, as well as at learn.eextension.org. Okay. So to introduce today's presenters briefly, we'll be hearing from Troy Gilmour, who is an assistant professor in the Conservation and Survey Division, School of Natural Resources, University of Nebraska-Lincoln. We'll be hearing from Vasu Sharma, who is an assistant extension professor and irrigation specialist at the University of Minnesota. And last but not least, we'll be hearing from Kevin Masaryk, who is a groundwater education specialist in the University of Wisconsin-Madison Division of Extension and the University of Wisconsin-Stevens Point. All right, so let's jump right in. Our first presenter is Troy Gilmour, and I'll just let you skim his bio there, see a little bit more about the experience he brings and where he's worked. And with that, we will get right into his presentation. So thank you so much, Troy.
Thank you, and it's really exciting to see so many folks online, and appreciate everyone for joining. I'll be talking about groundwater age a little bit, and this is a tool that can help us understand how aquifers impact stream water quality. Just some brief acknowledgements. I'll mention some of the projects that I have going at UNL, and I want to acknowledge students who worked on that. Collaborators past and present, North Carolina State University and University of Utah, and then funding from the National Science Foundation, Water for Food Institute, and USDA. So much of the water in streams and rivers originates as groundwater, and this cross-sectional view illustrates that process starting in the unsaturated zone, where water moves slowly downward below the root zone and eventually enters the groundwater system at the point of recharge at the water table. And of course, this water may contain elevated levels of nitrate from agricultural land use. And after recharge at the water table, that groundwater then starts to move downward and laterally toward the stream. And you can see that groundwater that originates close to the stream may have a short flow path, perhaps months before it arrives at the stream. And then there are longer, deeper flow paths where it may take decades to reach the stream. And so at any time, the stream water quality is impacted by a legacy of historical land use, including the nitrogen management, and not just the present nitrogen use. And so one other thing that is also important is that there can also be denitrification, or the natural removal of nitrate and conversion of it to N2 gas in this saturated zone before that groundwater reaches the stream. So this map is from a USGS study published in Environmental Science and Technology journal, and it highlights some of the areas in the US where there's a combination of high nitrogen loading at the land surface and where base flow makes up at least 40% of the annual stream flow. You see the base flow index is greater than or equal to 0.4 in any of the shaded areas. But the dark shaded areas highlight the areas with high nitrogen application and those base flow dominated streams where base flow is often attributed to groundwater seepage into the streams. And so one of the major questions then is, how long does it take those inputs to the land surface? How long does it take them to reach streams? And that's where this groundwater age tool can be of use to us. So at a much smaller scale, here's a cross-sectional view of a stream bed. What we're able to do in our work is to actually use the stream or the stream bed itself as an access point or a sampling point for that groundwater that has gone all the way through the aquifer and now it's about to seep out into the stream. And we can actually push in a lot of cases, in sandy stream beds or permeable stream beds. We can push in probes that are stainless steel or maybe PVC, and as little as maybe 30 to 50 centimeters. And by doing that, and we have a little 5-centimeter screen on the end there, we're able to capture groundwater that is discharging vertically up through that stream bed. So here's just an example of one of my colleagues at UNL, Aaron Middlestedt, doing that kind of work. You can see a piezometer here that's pushed into the stream bed. Here's a view of a transect out in the Nebraska Sandhills, just to give you a sense of those transects across the stream, the way that we often sample it. And then here we were during my PhD project, which I'll talk about more, in North Carolina. Here we're collecting some age dating samples, which actually take quite a while. And so we just decided to sit down in this case to collect those samples. But I'll show you some results from this stream in the coastal plain of North Carolina to give you a sense of the type of information that we can get from this sampling. In a lot of cases, we're sampling in those transect patterns like I mentioned, but then several of those transects along the length of the stream. So here are two maps that are a result of that sampling in that coastal plain stream in North Carolina. The gray dots that you see here indicate points in the stream bed where we sampled, and then we've interpolated, and there's a bit of a stretched longitudinal look to these because we've added some anisotropy to that interpolations. But what we're seeing here, particularly if you focus on the map on the left, these are actual nitrate concentrations that we measured. And you can see that band of blue down the middle, which is lower nitrate concentrations in the middle of the stream. We see higher nitrate concentrations along the edge of the stream. And collectively what we see is, first of all, an orderly pattern, but also we're seeing a wide range, right, in nitrate concentrations in the groundwater that's seeping through this stream bed. And so that's kind of interesting. We see this wide range in variability in nitrate, but we don't really know exactly why we have those patterns. And one way that we can start to tease some of that out is to look at the patterns of groundwater age. Again, groundwater age is the time between recharge to the time of sampling. So the travel time of groundwater between recharge and the time that we sample it. And in this case, we're sampling it basically at the exit of the aquifer, right at the stream bed, or just below the stream bed. But the takeaway from this map is that you see in this case, the red coloring, kind of this red band down the middle of the stream, is actually your older stream, or your older groundwater that's entering toward the middle of the stream. And you have younger groundwater entering toward the edges of the stream. And so if we flip back to this cross-sectional view, you can see that those patterns of deeper, older groundwater emerging in the center of the stream and younger groundwater with higher nitrate for more recent land use are actually entering toward the edges of the stream. So if you just go back and look at those two maps, you can see that pattern. Even though that illustration that I have is highly idealized, this particular stream, which was a kind of a channelized straight stream in the coastal plain, actually picks up on those patterns quite well. And we've done work, in addition to just mapping this and visualizing it, we've also done work because you've brought in that groundwater age component, you've now brought in a temporal component, right? And so we can actually use that same exact data that I showed you along with groundwater discharge measurements at each of the sampling points, and we can actually start to predict future nitrate discharge from the aquifer. And this is actually something that was pioneered out at UW Stevens Point back, I think it was published in about 2005 and it was worked in the sand plain streams by Bryant Brown. So anyway, these are things where we can start to look not only back and understand what has happened in the past, but we can also start looking forward because of the temporal component. So you might wonder how we do the groundwater age dating, and I'll just give you a very quick example. In short, we have monitored, we being NOAA, has monitored, for instance, chlorofluorocarbons in the atmosphere since 1950 or so. And because we know there's historical atmospheric trends, we can measure CFCs in groundwater. We can do some calculations to convert those CFC concentrations to an equivalent atmospheric concentration. And then we can come over to this plot, and we come over here for this particular sample, say it has that concentration. We drop down to 1980, and we say that groundwater was probably recharged because it fits that same era as far as atmospheric concentration is roughly 1980, maybe plus or minus 3 years or plus or minus 2 years, something like that. So we say that's the recharge year, and then we subtract from our sampling date. So if we had sampled it this year, we would say that water is, what, roughly 40 years old. So that's a little bit of the sense of how we get groundwater ages in practice. And so what we're doing next to try to move forward is thinking about upscaling beyond just the stream reach and thinking about much larger scales. And so here's an example of a small watershed in northeast Nebraska, which has high nitrate concentrations. I know that you can't see the vertical scales here, but we have nitrate on the vertical, and we've monitored the stream water quality under base flow conditions for just about a year now. And so what we're trying to do here on frankly a fairly limited budget is we've decided let's monitor the stream water quality over a period of time, and then we're going to bring in a small amount of age dating in these small green shaded areas, basically one 5-point transect at each site, and then we can bring in kind of a survey of the transit times of groundwater in this area that'll help us understand better why, for instance, this particular tributary has a lot of variability over time. Maybe it's a little bit more surface water dominated, whereas Spring Creek up here, as the name might suggest, seems to have very constant groundwater inputs resulting in stream water concentrations around 15 parts per million across the entire year. And so just one more slide here to give you some additional ways that we're trying to move forward in upscaling. We have a National Science Foundation grant out in the Sandhills where we have 4 intensively sampled reaches over 100 kilometers of stream length. And we're really working toward integrating that information with numerical models. And this will give us a much better reach spatially and help us to have a more integrated look. Now, of course, the sandhills don't have the nitrate issues, but hopefully we can learn lessons to take to these other nitrate-dominated areas. And so with that, I'll just leave you this map and this website that you can go to. It's an interactive website. If you click on the red dots, it'll pull up an actual study that involves groundwater age. And we're looking forward at these yellow stars to add our own studies onto this map in the future. So thank you very much for your attention. This has been exciting to speak to everyone.
Excellent. Thank you, Troy, for introducing us to the research that you've already done and some research that's coming up. Very helpful. Okay, on to our next presenter, Dr. Vasu Sharma. Again, an assistant extension professor and irrigation specialist at the University of Minnesota. So again, I know you can all read, so I won't read that to you word for word, but a little bit more about Vasu and the experiences she's bringing. So part of our purpose in these speed networking webinars is to introduce you to great people doing this work across the region, as well as the kinds of things that we're doing. So you know that Troy's there, you know that Vasu's there, and can follow up with them should you have any additional questions. Also, before Vasu starts, I'll just remind everyone that we're going to be saving questions for folks, for our presenters until after all three presenters are done, and you can go ahead and submit those questions in the chat box. So with that, Vasu, go ahead.
Thank you, Rebecca. I'm very honored to have the opportunity to speak here today with you all. So today I'm going to talk about the irrigation management in central Minnesota and how it is connected to groundwater quality. So here is the outline of my presentation today. First, I'll talk a little bit about my background, and then I'll talk about the current status of groundwater in Minnesota and how it relates to irrigation management. And then at the end, I'll talk a little bit about my ongoing research projects. So the projects that I'm going to talk to you about today are initiated this summer, so I do not have any concrete results to show you at this point, but I'll be talking more about methodology and some preliminary data that we collected this year. So I joined as irrigation specialist at UFM last year in July 2018. And before that, I completed my grad school, my master's and PhD in biosystems and agricultural engineering from University of Nebraska-Lincoln. And before that, I did my bachelor's in technology in agricultural engineering from Punjab Agricultural University in India. Talking about the groundwater status in Minnesota, we sure do have abundance of it at this point. Groundwater, in fact, is a primary drinking source for most of the Minnesota and provides almost all water for irrigation. However, the availability of groundwater varies throughout the state and is primarily the function of state's geology. So this map here shows you the location of 4 major groundwater areas in the state, and within each area, characteristics of groundwater are somewhat similar. For example, if you look at the northeast Minnesota, the bedrock is at the land surface, while in the southeast, the bedrock is 100 feet below the surface. So the availability of groundwater is based on that. In terms of irrigation management, the major area where the groundwater and agriculture are at conflict is the central sands region of the state. So this area, the central region, is characterized by coarse textured soils formed from glacial outwash, and groundwater is available from near surface glacial sands. Since the aquifers are very shallow in this region, the groundwater in this region is directly connected to lakes, streams, and wetlands. So because of this connection of groundwater and surface water, the leachate from the agricultural fields can easily enter into the groundwater wells, and because of greater pumping from these shallow wells in the peak irrigation periods, reduces the water levels in nearby streams and lakes. So according to Minnesota Pollution Control Agency, only 37% of streams meet water quality standards for aquatic life in this region, in the central region of the state, and only about 40% of shallow wells, which are less than 30 feet deep, have higher nitrate concentration for drinking water. What I want to show here is that irrigation in this area has a direct impact on groundwater quality. But at the same time, irrigation is very essential part of agricultural economy in the region. And as I mentioned in the previous slide, this area has mostly sandy soil. So the lands are very unproductive without water in this region because of very low water holding capacities of these soils. According to Department of Natural Resources, about 650,000 acres of land in Minnesota is irrigated, and most of this is in the central region of the state. So in this map, you can see the green dots here are the crop irrigation wells, and they are mostly concentrated in the central part of the state. And crops like corn, soybean, edible beans, and other canning crops are the major crops, with corn being the majority in this region. So this summer in 2019, I initiated 2 new research and demonstration projects at 2 locations in central sands region of the state. This map here shows you the location of 2 sites on annual precipitation gradient throughout the state. So these 2 sites have different soil types and different rainfall patterns, and are in the region where mostly the irrigation wells are concentrated. So in this first project, we looked at the interaction of irrigation and nitrogen fertilizer. Why we are doing this research and why it is important is because in this area, most related researches so far are focused on developing best management practices for nitrogen fertilizers and other fertilizers. However, the importance of reducing irrigation water or efficient irrigation management has been ignored or not studied rigorously. And also, many studies, if you go to literature throughout the United States and other parts of the world, have shown that moderate deficit irrigation conditions, under those conditions, a reduction in nitrogen fertilizer input does not decrease crop yield. So we thought deficit irrigation could be something that can be used in Minnesota as well in order to reduce nitrate leaching problems. But at this point, what is this moderate deficit irrigation level, and how much reduction in nitrogen is optimum for that deficit level is not known for Minnesotan Central Sand region. So the aim of this first study that I'm doing is to examine whether nitrate leaching can be reduced by deficit irrigation management, and then develop some irrigation and nitrogen combination best management practices aimed at maximum crop production and minimum nitrate leaching. The other objectives of this study are to estimate water and nitrogen use efficiency, and also estimate evapotranspiration and crop coefficients for corn, which are actually missing in this region for irrigation management. This year was the first year of our study at the Becker station, which is the East Central location. However, for our Westport site in the West Central region, we are going to establish plots in spring 2020, next year. The other approach in addressing the water quality issues that I mentioned in Minnesota is to understand irrigation scheduling. So the other research I'm doing is comparing different irrigation management methods, ranging from soil-based to evapotranspiration-based irrigation scheduling, in order to assess the influence of irrigation scheduling methods on total irrigation amounts applied, and then nitrate leaching and corn yield. So for our irrigation nitrogen interaction study, we are evaluating 4 irrigation levels ranging from 100% irrigation to rain-fed, and then 6 nitrogen rates ranging from 0 to 350 pounds per acre. This image here is the normalized difference red edge image that was taken on July 15th this year. And you can easily see the experiment set up here in this figure. The big green box is the main irrigation plot. and within each main plot there are 6 N rates. Since this image was taken on July 15th and only 1/3 of the nitrogen fertilizer was applied at this time, so you cannot see differences between nitrogen treatments, but still we can see no nitrogen plots which are highlighted in these red boxes. So we took these images later on throughout the growing season, but haven't analyzed it yet. Throughout the growing season, we measured soil moisture using neutron probe on weekly basis for irrigation management, as well as to calculate seasonal evapotranspiration under different N rates and irrigation management practices. The precipitation data for irrigation and obviously for water balance calculations for ET measurement were obtained from the weather station that was installed on the site by Minnesota Department of Ag. And for measuring the nitrate concentration in the water that is going below the zone, we used suction cup lysimeters that are installed at 4 feet depth. So we weekly water samples from these lysimeters and tested them for nitrate nitrogen concentration. We also took plant samples at tasseling and sorry. Yeah, so we also took plant samples at tasseling, and we are going to take plant samples at R6 growth stage in few days to evaluate the effect of these irrigation levels on nitrogen uptake. And then we're also going to collect soil samples up to 4 feet depth for nitrate determination after harvest. That would be in a couple of weeks, hopefully. So these two graphs shows you the total soil moisture in the root zone at 100% and 75% irrigation levels from mid-June to mid-August this year. And as you can see, the soil moisture in the root zone is always between the field capacity, that is the black line on the top, and management allowable depletion, which is the red line, in 100% irrigation, whereas in 75% irrigation, this moisture went below the red line, showing soil moisture deficit. So how this soil moisture deficit will now impact crop yield is the question, and will be soon revealed once we have the crop yield results in 2 weeks. However, this season was exceptionally wet, so I'm not sure what kind of results we'll get this year. Here, these are the two graphs showing nitrate nitrogen concentration in soil water from our suction cup lysimeters. So the graph on the left is 100% irrigation, and the two lines on this graph are the two nitrogen rates, 140 and 280 pounds per acre. The right side is a similar graph for 50% irrigation. And as you can see, we do see a difference in nitrate concentration between two N rates as well as two irrigation. So if you compare these two graphs, on average, nitrogen concentration in 50% irrigation was around 2 to 3 milligram per liter less than 100% irrigation at both N rates. And also, if we pay more attention at the irrigation season from mid-July to mid-August, there's a slight increase in N concentration in 100%, whereas 50% was quite stable. But as I mentioned, these are the preliminary results. We have to do more analysis on this data, and also going to calculate the nitrogen loads. The other study that I started compared 4 irrigation management strategies, so soil moisture and some ET based, and then epic growth model, to see how these models or predictive modeling can help irrigation scheduling. And based on these irrigation methods, we decided the irrigation amount and timing. So this season, the highest irrigation was in soil moisture plots, and the lowest was in irrigation management assistant tool. And we also did water sampling to suction cup lysimeters and compared all these 4 irrigation treatments. So now the yield results will show us that how these irrigation management strategies impacted crop yield, and then at the end nitrate leaching. So I'm looking forward to crop yield results now and see how these treatments are going to impact. This is all I have. At the end, I want to thank Minnesota Corn Research and Promotion Council and Department of Ag for for funding these projects. Thank you.
Thank you, Vasu, and we look forward to hearing more about the results of your research when you're ready. Okay, our final presenter today is Kevin Masaryk, and Kevin is joining us from the University of Wisconsin-Madison Division of Extension and the University of Wisconsin-Stevens Point. And Kevin does a lot of work in groundwater research and outreach, and we'll just turn it right over to Kevin.
Thanks, Rebecca. A little bit of background on me. I'm a groundwater specialist. My extension programming focuses mainly on rural landowners who rely on private wells as their primary drinking water source. Our extension here does a lot of work with them to organize kind of community-based testing programs where we offer them a convenient opportunity to test their water for nitrate and some other common issues or common problems that private well owners experience. So that's our main focus, is making sure those individual well owners who are on private wells have the information they need to determine the safety of their water supply. But through those efforts, we amass or collect a large amount of data, and we're able to utilize that data to help educate homeowners as well as communities about the impacts of, let's say, land use activities on groundwater quality. So I'm going to share some of the tools that we've put together and use to educate people about those concerns. So just a little bit of background. We often hear that we don't know anything about nitrate in Wisconsin's groundwater, and I like to kind of prove people wrong. We have about 800,000 wells in the state of Wisconsin, and over the last 30 years, extension efforts have collected data on over 100,000 wells. There are other entities such as our Department of Ag, Trade and Consumer Protection that collect data. Wisconsin Department of Natural Resources collects data as well. And as of 2014, the state now requires that all new wells or wells that have well or pump work done requires that those wells be tested for nitrate and bacteria as well. There's also a host of health departments that might have their own labs that also collect data on those certain parameters. So over the years, we've collected that data, and then we've created a tool that allows that data to be aggregated. It's great that people determine or find information about their own individual well, but the next question is, what do we know about groundwater quality in my community, or how does my community compare to others around me? And that's where the Wisconsin Well Water Viewer comes into play, is that it collects and aggregates that data and provides it in a way that is, somewhat transparent. We never release location of individual wells necessarily, but we do aggregate that data that allows communities to see what is known or what isn't known about water quality where they live. So that's a resource that's available online, and I just want to do a quick overview about some of the functionality that's in there. This is a map showing it's showing the Wisconsin counties. The different colors correspond to the different concentrations, average concentrations of nitrate by county. You'll notice on the left-hand side that there's other parameters listed. Nitrate's one of about 14 or 15 parameters that you can select and view data for. Nitrate is the most extensive just because it's a relatively inexpensive test and it's got health concerns associated with it, so it's one of the more common tests that people over the years have performed. County is great, but sometimes doing an average by county masks or doesn't tell the whole story in terms of water quality in an area. Groundwater quality can obviously be much more localized. So we do allow people the opportunity to select or display data by different scales. So this particular map shows data aggregated at a town range level. And now we see some blank areas which indicate areas where we haven't collected sufficient data to characterize the average concentration for that region or those areas. And that's usually a function of just not a lot of wells, not a lot of population to draw from. The other feature that we have is displaying those concentrations of nitrate by different colors. Some people have trouble seeing different or certain color schemes, so the ability to select different color ramps maybe makes it a little bit easier for somebody who has trouble perceiving color. It makes it easier to see the differences in this map. We also have the ability to zoom in to the section level. So this is the most detailed level that we would display information at. And there's a lot more blank area showing up, but there are quite a few areas of Wisconsin where we do have fairly good coverage that we can color code most of the county at this level. And then you'll really begin to see or give people the ability to see how water quality varies across a county or across the area where they live. Another feature that often is a little bit hidden is that you can change the base map. So when we're trying to educate people about the relationship of land use to water quality, obviously seeing an aerial photograph is a very visual way for them to understand what the underlying land use is. So by changing the base map and allowing you to adjust the transparency of the layer, it gives people the ability to kind of understand those relationships a little bit better. Another function that exists is you can actually define a user-defined area, and once that area is defined, it will generate a table showing you a little bit of additional information on the underlying data set. So breaking it down by a range of concentrations, summarizing the statistics, the maximum, median, average, minimum concentrations, as well as giving a percent exceedance rate. So what are the percentage of samples that exceed the drinking water standard of 10 milligrams per liter? And there are different layers that you can even generate statistics for. So if you wanted to generate a table based on county, it would allow you to do that. In this particular example, it allows you to select various watersheds, and it'll summarize the information or what is known about nitrate for that specific watershed. So that's some of the functionality that have been built in. This is an Esri product, so it's kind of an off-the-shelf Esri product with some ability to customize on some level, but most of the features in here are kind of built into that product and used to display the well water data for the state of Wisconsin. So that's a little bit about the well water viewer. Just transitioning into what communities want to know from that information. It's often the case where they want to know what is impacting our groundwater, what's causing our groundwater to be impacted. And that's where utilizing this data and comparing it to other available data sets really comes in handy because it does help paint that picture of water quality in the state of Wisconsin. And nobody in this room is probably surprised by the fact that nitrate concentrations in groundwater are a function of land use, but also the soils and geology, which can exacerbate nitrate losses to groundwater, really do paint that picture. And tools like this provide the ability to tell that story to those residents or those citizens of states or regions that might not have the background or the resources to understand all aspects of nitrate loss to groundwater. So these tools really do serve as a resource for people like myself, but also other professionals, local health departments, to really start educating people about the impacts of land use on water quality, but also giving people a better understanding of where to go for certain pieces of information. So that's at the statewide level. More locally, there have been counties where we've done more detailed sampling or more detailed analysis. This is an example of Chippewa County, which is in northwestern Wisconsin, showing that when we have nitrate data, and we have data on, let's say, well density or septic density, when we have soil drainage classification, when we have good land use layers, we can then develop statistical models that can be used to help predict things like nitrate concentration in that area. So by utilizing that data, utilizing other data sets, we're able to better help communities understand where nitrate is likely to be a concern, where it's not. And that just helps us focus our outreach efforts as well as our conservation efforts. This is just another slide showing actual data on top of our predicted value, but just showing some of the capability of the tools that we implement to help communities in Wisconsin. So with that, that's all I have, and I'll turn it back over to you, Rebecca.
Great, thanks so much, Kevin. Okay, now we are finished with our three presenters are finished with their presentations, and we will move on to questions from our participants. So our first question is from Harbins 4, and he asks what, and I think this was for Vasu, what does different levels of irrigation mean? I think you referred to different levels of irrigation. You had a couple of different slides that illustrated those levels. So could you just say a little bit more about those different levels of irrigation?
Yeah, sure. So 100% irrigation, from 100% irrigation, I mean is we replace all the water that is being removed from evapotranspiration. So we use soil moisture sensors. So this is soil-based irrigation scheduling. So we use soil moisture sensor data in order to know how much soil moisture is left in the profile, and based on available water holding capacity of our soil, we every time our moisture when approaches that allowable condition, which is 50% of our available water holding capacity, we irrigated to refill that profile up to close to field capacity. So that is our 100% irrigation, to bring back our moisture back to close to field capacity. And then our reduced levels, which is 50%, 75%, is just the reduction from 100% irrigation.
Excellent, thank you. And Nicole from the West Polk Soil and Water Conservation District in Minnesota asks, how or who did you contact to obtain the number of irrigation permits?
So this data is online I took this from the DNR website, and it's present there. It's public data that you can use, and you can also download the ArcGIS layer for irrigation permits from that. But the picture that I used in my presentation was from one of the presentations from DNR, and there's a link at the bottom of that picture that you can use to reach there.
Great, thank you. And another question for Vasu from Andrew Craig here in Wisconsin. Your irrigation study showed two nitrogen rates, 140 and 280 pounds of nitrogen. His questions are, first, was this for sweet corn or another crop? And second, what's the University of Minnesota's recommended N rate for that crop? And you can see the rest of his question there.
Sorry, I was muted and I was talking.
Yeah.
So the rates that I mentioned here, 140 and 280 pounds per acre in my slides, is just an example. But we looked at different rates starting from 0 to 350. So for the sake of this presentation, I just used two levels. One is below the recommended and one is above the recommended. And the field corn was the crop that we used. So we are doing this for continuous corn cropping system. The recommended nitrogen rate for irrigated corn is from UFM is not 140 and not even 280. It is in between these two numbers. It's around, I think, 210 to 250 for irrigated corn crop in the state. But for this presentation, I just used it as an example, one lower level and one higher level. If that answered the
Perfect.
question.
Yes, thank you. Jonathan, this is for Troy. For the nitrate dating on groundwater, how was the groundwater movement estimated? And he's saying there are areas in the aquifer where infiltrated water from the soil surface takes more than 5 years just to reach the water table, and more than that for the water to show up in the stream. So help us out, Troy.
Sure, and Jonathan brings up a really good point, and probably something I should have emphasized more, but groundwater age is that time starting at the time 0 is at the water table. And so if you have a thick unsaturated zone or vadozone, then you may be missing, like Jonathan suggests, a significant time component in the overall time for a solute such as nitrate to move, all the way through that system, both the unsaturated and saturated zones, to a stream. One other thing is, I maybe should have emphasized too, is we often refer to these groundwater ages as an apparent age of groundwater, and that just kind of helps remind us that we're using imperfect tracers, basically. And so, but we are careful to call it groundwater age dating as opposed to, say, nitrate dating. There is other work where folks have been modeling, for instance, the recycling of nitrogen in the root zone and the time that it may spend there before it actually leaves that area and moves on from the root zone, for instance. So this is something that we specify groundwater age dating. Just one additional thing is that there's a little bit of work being done in this area in Nebraska, because we do have areas with thick vadozones. And I'm involved only peripherally, but Dan Snow is one of the people who is central in the work, and there's a lot of vadozone coring being done, and then nitrate being extracted, and seeing nitrate levels in that soil water or in that vadozone area, and getting a sense of the mass of nitrate that's still on its way down to a water table, and still, like Jonathan mentioned, may take years or even decades before it even reaches the water table. So yes, that's a really important question. I appreciate it.
Okay, thank you. And just a kudos to Kevin for your team in making a more accessible map using that color variation that you've used. And then Kristen also has a question on the well water viewer. She says, looking at a township scale, is there more information regarding the depth of groundwater referenced for the nitrate level?
So I'm guessing that it's in reference to, do we know anything about the depth of the wells, and the nitrate concentration as a function of depth? And that is currently one limitation in the viewer, is that it aggregates a bunch of information. Historically, wells in the state of Wisconsin, they do have well construction reports, but a lot of times that data doesn't get really good until about 1988. And it's only good when people submit their Wisconsin unique well number. So as we move forward, I think with some of the statewide requirements to test wells at the time of construction and anytime there's well and pump work done, we'll be able to do a better job of relating nitrate concentrations to specific well depths or specific depths below the land surface or within the aquifer. But at this point, that's a little bit more aggregated data and doesn't allow us to assume much about the vertical component of nitrate in groundwater. What I would say, however, is that a lot of times wells are drilled to similar depths or similar specifications depending on the area you're in. So I do think it does a pretty good job of kind of painting the picture of average well water quality in a region. However, I would acknowledge that our ability to understand vertical penetration of nitrate or vertical differences in nitrate concentrations in an aquifer might be a little bit limited by the well water viewer's current functionality.
Okay, thanks, Kevin. And Kristen has a follow-up question there, asking if all samples tested are tested for nitrate, or are all samples tested for nitrate at UW Labs added to the well water viewer?
All samples tested at the Water and Environmental Analysis Lab at UW Stevens Point are added. There are other, county health departments that have voluntarily submitted their data sets to us, but there's a lot of other labs that have not, and it's currently a function of kind of staff, and our ability to integrate, many different data sets is a little bit limited. So we've done that on a pilot scale. In the future, there might be opportunities to do that, but currently it's mostly limited to a few labs, which still has amassed, a fairly large data set that isn't complete, but is enough to kind of give us general indication of groundwater quality throughout the state.
Okay, thank you, Kevin. And a secret sauce question from Erica, who is asking, how are you able to test so many wells? And saying they've been trying to do something similar, but are having a hard time getting people to submit samples, even with free sampling.
Yeah, I think probably the greatest barriers to testing are for people not knowing what to test for and not knowing where to send a sample. Cost is obviously a limitation to some folks, but I think not knowing what to test for, not knowing where to send a sample are some of the biggest barriers. So Extension has a long history of working with communities to try and overcome that through localized testing. Through those efforts, we might collect 3,000 to 5,000 samples a year. And over the last 20 to 25 years, that amasses or amounts to a lot of samples. But it is worth noting that it's taken a long time to get to that number. Just in the last 5 years, however, I think the state of Wisconsin, through their requirements to require new wells to submit nitrate samples, as well as any well that has well or pump work done, that's allowed them to collect about 60 to 80,000 samples just in the last 5 years alone. So voluntary efforts, I agree, it can be a little bit challenging. That's where, requirements or changes to regulations to collect some of that data can also be useful or handy.
Okay, thanks, Kevin. I'll ask a question of Troy. Troy, can you give us examples for how groundwater age might be used or is being used to inform management decisions, either at the farm or property scale or county or the NRD scale for you all?
I guess like a first broad pass at that question is that one of the things the groundwater age dating has done, and it's been done for some time now, is kind of help people get a realistic time frame in mind for the impact of regulation, changes in management practices. And so if you realize that it may take decades to flush, nitrate out of an aquifer, then that changes your thinking about, for instance, how long it would take a stream water quality to change in a groundwater dominated stream. So that's kind of a big picture answer to your question. More specifically, we're doing that. The one project I mentioned in Northeast Nebraska is in a very special groundwater management area called the Brazil Groundwater Management Area. There's EPA funding associated with that area, and there are 4 natural resource districts that all kind of intersect in that area. And so we're hopeful that, collectively the surface water sampling and the groundwater, a little bit of groundwater age dating there will help us kind of get a sense there as well kind of what kind of time scales are we talking about here, and what can we maybe expect in the future?
Okay. All right. Last chance for questions, folks. If you type fast, and while I give you a chance to do that, I'll just move on to our final slide. And thank you all for participating, and thanks so much to our presenters, Troy and Vasu and Kevin. Great mix of work here. And this, as I said, this session is recorded and will be available for folks that were not able to make this time. So if you know anybody that you think might be interested in the information, please do pass these links along. Our next webinar is brought to you by our North Central Climate Collaborative. It's a sub-team of the North Central Region Water Network. And that next webinar is about climate change's effect on field crop diseases. That will be Monday, October 28th at 1 p.m. Central Time. And you can find information on that at northcentralclimate.org/webinars. So thank you all for joining us, and look forward to having you at our next webinar.